these lectures very inspiring and informative. Now, starting with the day, I would like to introduce you all to our today's speaker, Dr. Pamita Avasthi. Therefore, I request ma'am to kindly deliver her talk. Thank you. Ma'am, your mic is off.
Good morning to all the attendees of this online 1 week yarn program. Uh, it's a last date, indeed, it's a last day, and I'm pretty hopeful that uh, all the lectures, means you might have benefited from all the lectures. And on these concluding days, my lecture would be on application of G quadruplexes in chemistry and nanoscience. And much has been covered. So I'm just trying to conclude what had been discussed by the other speakers and how this technique is being applied in a real sense, and what is the level of research worldwide going on. So, am I audible? I think I switched off the speaker also. Am I
Yes,
audible?
ma'am. Yes, ma'am, you're audible. And the
Okay,
screen is also visible.
okay. So, DNA quadruplexes, they have a diverse application in biotechnology, pharmacy, pharmaceuticals, biomedical sciences, material science, and engineering. These structure formed by guanine-rich DNA sequences, as we had discussed, just a minute. So these sequences, the guanine-rich DNA sequences, they are being explored for their potential as a therapeutic agents, diagnostic tools, and also in the development of the noble material. So I will just conclude my first lecture. In first lecture, we had discussed about the primary, secondary, tertiary, quaternary structure of the DNA. And we had also discussed that how the secondary structure is responsible for the polymorphism in a DNA, and which has a different application at the cellular level. So if you see the one of the form of the DNA, it is the quadruplex structure, where the four guanine bases, they arrange themselves in one plane with the Watson and Crick and Hookstein hydrogen bonding, and in totality you have 8 hydrogen bonding among us this four guanine nucleotides, or you can say the nucleic acid bases. And this is just a representative a representation that how they are being arranged. And this is the phosphate backbone, which is also called as monomer. And they are being arranged by stacking one above the another. So whole of this structure is being called as a G quadruplex. So, and the stabilization, one of the major stabilization factor is that the presence of these ions, either it is a potassium, sodium ions in the center, which is coordinating with these four oxygen atom above and below. So all these things we have discussed in our very first lecture. In this lecture, obviously, as I would be discussing the application, so it has a very diverse application. So I have categorized into a two, that the in medicines as well as in therapeutics and biotechnologies as well as in material science and engineering. Though I cannot take all of them because lot and lot of work is going on worldwide in these areas, and the plethora of information is available online, offline, in print media, everywhere this is available. So, if you start one by one, just brief you, but I will take these two, cancer treatment and biosensor, a little bit in a more explanatory way with some example. And our laboratory is working in this particular area, that is a cancer treatment, taking quadruplexes as one of the binding or receptor site. So quadruplexes, they are attractive agents for a cancer therapy, and ligands, they are designed to target these structure as they are being considered to be a very inherently or you can say the integral part of the cancer cell growth. So by allowing the ligands or new molecules to interact or interfere with the DNA, and ultimately these quadruplexes, or we can say interfering with the gene transcription or replication processes, so which is all being controlled by the DNA. Quadruplexes, they can be used to enhance the drug delivery by incorporating them into the nanoparticle or in hydrogel carriers. As a diagnostic tools, the quadruplexes can be used to enhance the drug delivery by incorporating them into the by using them as a biomarker for various diseases. So this unique structure of quadruplexes allow them for the development of very very sensitive, or you can say a very specific diagnostic tool. In gene regulation, of course, they play a very vital role by targeting them, and they can act as a very, or they can have a very potential use to modulate the further gene activity in specifically the therapeutic application. G quadruplexes, they can be used or they can be exploited in designing as an antimicrobial agent by targeting their information in the pathogen. So I think if you have attended the Professor Berthwal's lecture, or if not, please go through her lecture. She has very well explained that the quadruplexes structures, they have also been found in the viruses as well as the bacteria. So this is one of the very important application. Now, in application in technological area like biotechnology, material science, and engineering, so they can be used as a nanomaterial to create dynamic DNA nanostructures, including those which have a switchable properties, and they can be used in catalytic functions as well. So, as a biosensors, the G quadruplexes can be used to detect the specific molecules or DNA sequences and make them valuable for biosensing applications. They can be used as a biocatalyst and can be engineered to act or facilitate the chemical reaction in a very controlled manner. Similarly, as a hydrogels, they can be developed for various application, including cell culture, tissue engineering, and drug delivery. So overall, what I would say is this is a versatile class. So G quadruplexes is a versatile class of nucleic acid structure with a wide range of potential application. It is their ability to form a stable self-assembly structure and also their role in critical cellular processes which make them attractive target for research and development in various fields, right? So I would be taking two application, the cancer treatment and the biosensor, from these two broadly categorized application area of G quadruplexes. Hello, I'm audible. Hello, am I audible?
Yes, ma'am, you are audible.
Okay, fine. Chal. So then, Let us start for that how these quadruplex structures, or what kind of information, or what we can do with these quadruplexic structure in cancer treatment. So, as we discussed in first lecture, that we discussed like primary, secondary, tertiary, and quaternary structure of the DNA, and they are so long that definitely they require a specific mechanism to get folded inside the nucleus in the form of a chromosome. So whole of this process is called as a supercoiling, and variety of or various proteins or enzymes, they act upon this DNA to unfold it. So the question comes is that what is the role that is being played by these quadruplex forming sequences in the genome, right? This is a complete genome which is capable of forming the quadruplexes. So till date, it has been observed that the telomeric junctions, they are the presence of or I can say the presence of G4 quadruplexes has been observed in this particular area, that is in telomerase area. So and they are but in a very latest research, it has been proposed that yes, the non-telomeric G quadruplexes, they have also been observed. So what exactly, in order to find out their function or their role, the answer is that function of these quadruplexes, you see this part formed in the promoter region of the oncogene CMYC, right, which results into the down regulation of gene transcription. So this is the promoter area in a gene where all the transcriptional or translational activities it starts. And if some kind of a mutation is being sensed, so this has been observed that quadruplex, they disrupt all of this process and would lead to the apoptosis. So, and this function they say that this function is being, or researcher says that this function is achieved by acting as a steric block to the transcription processes, either directly or as a ligand to which the protein can bind and block the transcription processes. So this is what has been observed, and the role which is being played by the quadruplex. So we have discussed this structure in detail in our first lecture, that how the quadruplex, what type of different quadruplexes they are in different type of structures that has been solved by X-ray or NMR crystallography. So next question comes that how do we confirm? Okay, we have identified it. How do we confirm the presence of quadruplexes in our DNA? So this is something cellular studies in which the ligand treatment that has been done, or you can say that in order to confirm the G4 structure, the first ever in vivo experiment using G4 structure is being done by specific antibody to stain the G4 in the telomeres, whereby the telomere structures formation was observed to be dynamically controlled through the protein interaction in a cell cycle dependent manner. So when whole of this process when you have to identify their position, you will expose the cell to the specific antibodies. Specific antibodies, what I mean to say, which will bind to these quadruplex only, not to the duplet or not to some other segment. And position of those antibodies which are specific to this quadruplexes that has been identified by various cell cycle dependent manner experiments. So these are some of the experiment which has been done in series to confirm the position of quadruplexes on the genome. So one of the important experiment is the chip sequencing. This chip sequencing. So what does this chip sequencing mean? Is this is chromatin immunoprecipitation, which is commonly used to detect the interaction between the protein and the DNA in a native chromatin context. Further, in combination with high throughput DNA sequencing analysis, it allows genome-wide detection of protein binding site. So this is specific. This particular technique is used to detect the interaction between the protein and DNA. So this particular technique or technology is exploited here to detect the antibody binding against the G4 structure, right, rather than the protein. So this technology over here is being used to detect these structure instead of protein. So lot of our combination of experiment, that is the fluorescence microscopy or simple G4 sequencing or permanent net footprinting or chip sequences of G4 binding proteins or markers or simple G4 chip sequencing. So all these experiment at the cellular level confirms the position of G quadruplexes on the genome, right? So G4, they are not randomly, as I told you in the beginning, that they are not randomly distributed throughout the genome. So there are the enriched region on the genome, such as the promoters or telomers or transcription factor binding sites where the formation of G quadruplexes has been confirmed. So, by all these experiments, right? So, about 40% of the human promoter region has one G4 motif. So, different biochemical and molecular experiment underlines the presence of G4 structure where they support or affect various biological pathways. So, for example, in protein expression or in telomeric activities or in form of genome stability. So, these chemical experiments they confirm, right, the formation of these G4 structure, specifically in promoters in telomerase region of the genome. Further, how does it next question arises, that how do we relate it to the cancer? So experimental studies provide a proof that increase in G4 structure in the cancer cell in comparison to the normal and the healthy state of affair of the cell, the G4 activity is or you can say the telomeric activity in the cancer cell is maximum, or you can say there is an increase in a G4 structure formation in the cancer cell. So this is the cancerous cell, And you could see this is the normal means these are the G4 strands. So these type of structure formation in the cancer cell is more. So therefore, based upon these cellular studies, right, this G4 formation that can be used as a anti-cancer treatment strategy by blocking the replication or blocking the oncogene expression. So, taking all this study, what I've discussed up till right, the this a lot and lot of or thousands of ligands, they are being designed to block the activity of these or formation of these quadruplexes in the cancer or in the cancer cells. So that can be done by controlling the telomerase activity or going for a telomerase stability, or by reducing the oncogene expression in the form of a transcription or translation, or controlling the transcription or translation, or by controlling the genome instability to counter the mutation, or promote the apoptosis process at the cellular level. So undivided cell means replicative immortality, which leads to the cancer, right? So which protecting the or by protecting the proliferation of the chromosomal end. So number of molecules, they are they and their structure, or they are being proposed or designed, and their structure with the four-stranded quadruplexes has been reported by X-ray and NMR spectrophotometric tools, which confirms, right? This is one of the structure, so which confirms the the binding of these molecule to these quadruplexes DNA. So I will not go into much details. Definitely, it is require another lecture, that how these molecule go inside and what kind of stabilizing and destabilizing effect and how it proceed to the side. I will just give you an idea that these are the various types of molecules which are being designed to interact with these quadruplex structures, right? And out of these, some there are number of it, as I said, thousand of more than 30 to 40,000 molecule library of molecules. They are being deposited to they are there in the data bank. There are some of the common which I have shown over here, very commonly used as a binder. So one of it is MM41, which is very commonly used. So this is the crystal structure of binding of MM41 with the DNA quadruplexes. So so how do we design these molecules now? So we design these molecule based upon the binding interaction of the one of its candidate with the receptor, and just in order to improve the binding, we just modify the structural feature, which are present around the main chromophore or simple chromophore modification. So these small molecules, or you can say the G4 binder, they have a aromatic surface for pi-pi stacking interaction with the G tetrad, and these side arms, which can be positively or negatively charged, or we're having some kind of a basic group, they falls towards the loops, or you can say the groups where they show some kind of interaction and ultimately lead to the stabilization. So, in order to improve this binding, the aromatic count, the positive charge, or number of the hydrogen bond added to these molecules, it has been proposed that their pharmacokinetic activity or property that also get enhanced. And based upon the structure activity relationship studies on G4 ligands, the if we increase, this is what they propose in order to control their physicochemical properties, such as the planarity, polarity, or lipophilicity, and rotation about around these bonds would able to balance the binding interaction with the G4 along with their solubility as well as the permeability. So this is what has been concluded out of the structure activity studies. So these are the two types of you can say um, quadruplexes, the parallel as well as the anti-parallel, or you can say the hybrid structures. So the binding of DR47 shows means in this particular case, what I'm trying to show is their preferential binding, whether they bind for a monomeric, they bind for a dimeric, or they bind for a tetrameric kind of a quadruplexes, or they bind for they're binding towards the hybrid kind of a structure where you have a mix of parallel as well as the antiparallel kind of a quadruplexes structure. So the two molecules, that is the DR47 and dinickel sulfene complex, the DR47, they show the preferential thermal stabilization of a G4 dimers, and they are showing a greater preference for dimer as well as trimer in comparison to the monomer. Similarly, if you compare this one, this type of this dinickel sulfene complex, they will show 34 time or 30 fold greater affinity for a dimer kind of quadruplexes in comparison to the monomer type of a quadruplexes. So dimer and monomers means that where there is a one nucleotide unit or one strand of DNA which is getting folded into a G quadruplex, and in dimer the two units, they are folding in the form of a quadruplex, two strands, and in case of trimer, three, and in case of tetramer, four. So the site which has been confirmed by the NMR and X-ray diffraction study is the stacking between the two sets of tetrads. This is the two sets of tetrad. It does not go here in between. It goes between the two sets of these tetrads, and having an interaction between these, these loops, or you can say these flanking nucleosides as well as the groove. So this is what has been confirmed by the structural study. So all these structural as well as cellular studies is, yes, important to us as a chemist, because they give us an idea that how do we proceed for further design and development of new class of quadruplex binders. So keeping all this literature into consideration, so our lab is working from last 15 to 20 years in the area of designing of anthraquinone class of compound as a DNA binders, whether it is a duplex or whether it is a quadruplex. So we have done a very comprehensive study on designing as well as synthesis of these class of compounds, as well as their cellular studies also on various cell lines, cell cycle analysis in order to check their target, and also the biophysical studies of these compounds on the various DNA sequences using UV visible fluorescence and other related spectrophotometric technique. So I will not go into that detail, because obviously that comprises the thesis, the 3 to 4 thesis work and one postdoc work. I will just conclude what exactly we are doing in order to check the structure activity relationship. So as I told you, we need a planar aromatic ring. This is a planar aromatic ring in order to have a pi to pi interaction with the DNA. And the another, and which has been confirmed by various studies from last 15 to 20 years, that it is mandatory in order to have a stacking interaction with the receptor site. The position on this ring, whether it is 1, 2, 3, 4, 5, 6, 7, 8, they play a vital role. So just by changing the position of the side arms, we have observed there is a drastic change in the bioactivity of the compound. And further, we have also varied. So this is the position, the red arrow is the position, blue arrow is incorporation of more sp3 factor in the middle of the chain, right? Or at the end of the chain. So this also contributed to its bioactivity in a real state of affair. So these are the number of compounds which are being designed as well as synthesized by simply, in this case, position is fixed, but we have changed, we have diversified the central component of the side arms as well as the terminal component of side arms. And the publications are also there, so you can check it online for the details. Further, now we have added another arm at the position number 5, and also the same variation we have done at here also at this position. And also there was only one arm. Here we have added another arm at the 5th position, and also checked its bioactivity on a various cancer cell lines, also cell cycle activity, and followed by in vitro biophysical studies. Then again, we have now, instead of one and as well as one five, we have chosen the position number 2. This position number 2, again the same thing. Now we are changing the position and also simultaneously changing the structural feature at the side arms also, and confirming its bioactivities. Similarly, now the position, additional position is 1, 4. So what we have done is, we have varied the structure at the side arm. We have varied the position also from 1, then to 2, then to 1, 5, then to 1, 4. So there is another student who is synthesizing 2, 4 substituted compound in order to check the bioactivity. So that work is under process. So this is the library of the molecules we have designed and synthesized, and we have checked their anti-cancer activity. So I will not go into that details, that the various type of anti-cancer, antibacterial studies we have done from on various cancer cell line as well as the bacterial cell lines. On anti-cancer cell lines, we could calculate the IC50 value, and from the IC50 value, we have picked up one of the best compound, which is in comparison to the standard drug, that is a mitozentron. Mitozentron is the the structural similarity with the mitochondrion is that it is also having anthraquinone and having same side chain on these position 1 4 position with a different structural feature, and also OH group at 1 or 4 position over here. So we just refer them as a structural analogs of mitoxantron, and hence we have used mitoxantron as a standard drug in order to compare its bioactivity with it. And to our surprise, that we have found that 2 to 3 compound out of this library has shown the better effect than that of a mitoxantron in some of the cancer cell line. Mitoxantron is showing bioactivity in all the cell line, but these compounds, just by wearing the structure feature around the chromophore, they are active to specific cancer cell line. And by doing the cell cycle analysis, we have found the major blockage in the cellular activity is at the S phase. S phase is where the DNA replication that proceeds. So, as per our understanding from the literature as well as from our study, so we could conclude that yes, these compounds they can be a good G quadruplex binders, though they are showing some kind of fall in cellular activity in a G1 phase also, which is the initial phase of for of the when the process of cell division starts. So they do active they show inhibition at that stage also, but at the S phase the maximum damage is being done. So we also this is something which we have observed from the cellular studies as well as its comparison with the literature. Then we have the comparative analysis, the biophysical studies of all these compounds in comparison to mitochondrion using absorption emission analysis, spectral analysis. And what we could confirm is out of these studies that yes, there are multiple binding site, but it is not a dynamic kind of means it is not a kind of a collision with the receptor, but it is a kind of a binding interaction with the receptor. So this is what I could conclude. So I cannot go into that detail by going one by one spectra, because all these things has been discussed in the previous lecture by Professor Barthwal as well as by Dr. Eliza. So I'm just showing you that what we could conclude. We could conclude yes, they go and they interact with the receptor side, that is the DNA, and they are showing a stacking interaction. And all these studies they are being done in comparison to the standard drug, that is mitoxantron. Similarly, we have also done for various analogs. So we have chosen one of the best analogs, and or I'm presenting. We have done it with all the analogs, but I'm presenting here the best analogs which are showing the binding interaction with the at the cellular level, and also in vitro these spectrophotometric titration. So as I told you, by confirming that yes, there is interaction and there is a kind of a binding interaction with the receptor side. So we also did, because the binding interaction of one of this molecule, which is coming out to be the best with the G quadruplexes, which is being a part of a human telomeric sequences, and we could confirm the same observation what we have observed at the DNA duplex. And we could see the binding interaction is better than the standard drug, that is a mitocentron, right? That is in and if you go to the binding interaction of the mitocentron with these quadruplexes, so we were getting the same result what is being observed in the literature. Similarly, the in case of same observation we have observed in the emission spectra also, and we could see the same trend what we have observed in the cellular studies as well as in the duplet iteration. We were quite adventurous, and we tried to find out, because it has been said that side effect of the standard the cytotoxicity of the mitozentron is because of the presence of these OH and these ketonic functionalities on the ring, and because of this they generate more and more free radical, and ultimately leads to the cytotoxicity. So that is the next part of the experiment, but we have used the another experimental technique, that is a cyclic voltammetry, right? Where we can we have also observed the same interaction or sorry the same results what we have, or the results that they correlate with the result what we have observed for the absorption as well as for the emission spectroscopy, and binding interaction with the quadruplexes has been confirmed by one of our best compound which is showing anti-cancer cancer activity. So we have been granted patent for these compounds. So this is what we have done, and this is the kind of work which is going on all over the world by the various group in order to design and develop a cancer or G quadruplexes targeted ligands, which is having a very wide application for the cancer treatment. So next area of application is the biosensor, or we can say that how these nano structures based G quadruplexes biosensors, they are being designed. So this is the structure of the thrombin binding aptamers, which is a 15 residue DNA oligonucleotide with a sequence of GG, TT, G means guanine, T means thymine, GG, TG, TG, GT, TGG 3 prime. So this nucleotide, or we also call it aptamer, that show a high affinity and selectivity to protease thrombin, which is an enzyme, or which is an which enzyme which is crucial in blood clotting. So nanomolar concentration of this DNA aptamer, they can inhibit the formation of the fibrin clots, uh, which results from the thrombin activation. So this single strand aptamer, they can form a unimolecular G quadruplex. So you can see over here. So this can form this type of unimolecular G quadruplex that is being shaped like a chair, right? You can see the two, one, two, three, four. This is one stack, another stack. So these are two stacks of G quadrat, which is connected by these two loops, T12 and T13, right? T3 and T4, and there is a central three-base TGT loop, right? So this is one loop. These are all edgewise loop, right? And this is a unimolecule. You can see unimolecular G quadruplex. So the potassium ion is important for the which is there at the center for stabilization of this structure. So NMR and mass spectrometry, they confirm the presence of potassium here, right? So till date, number of thrombin biosensors have been developed. However, later on, it got noticed that it has a potential to be used as a therapeutic anti-thrombotic agent, right? Further, there is, I can say that this nucleoside is also important in supramolecular chemistry for serving as a primary model for development of a range of sensors as well as the nanomachines, which I would be discussing subsequently. Further modification of these 15-mer oligonucleotide, or you can say the TBA aptamer, will fold into a G quadruplexes under a well-defined condition, which has been exploited to develop optical and electronic sensors, which range from potassium sensors to protein sensors to nucleic acid sensors. So variety of sensors, they are being designed using this as a base model. So I would be discussing all these things in my next slides. So G quadruplexes as a potassium ion sensors, right? So the modification of this TBA, that is a thrombin binding aptamer, as a fluorescent indicator for the detection of potassium ion in water, has been proposed by Tikenaka's groups, right? So attachment of these pyrene ring at the 5' ends, 5' end, and 3' ends, so at the two terminals of this oligonucleotide. So DNA, whole of this structure gave a probe, or you can we refer it as a DNA probe, which named as PSOPY, for potassium sensing oligonucleotide packed attached with the pyrene molecules. So PSOPY. So this is a primary sensor. This is being referred as a sensor for a real-time detection of the potassium ion in biological or in environmental sample. So one challenge is the selectivity of a potassium ion in the presence of sodium ion at higher concentration, and another challenge is the fast response for real-time monitoring of the interaction of cation. So how it functions? This PSOPY is used the excimer formation. I think excimer formation is very well discussed by Dr. Eliza, so I will not go into that slide. Maybe in day 2, she has explained that what is excimer and how the excimer form and what fluorescence technique with excimer formation. So if you would like to recall it, please go back, because all the lectures, they are recorded, so you can refer her lecture. So excimer formation for a pi stacked pyrene to single potassium binding. So, but in the absence of this potassium, this whole of this PSOPY is in the unfolded form, and they will not show any kind of excimer formation. So that is a fluorescent signal, I should say. So when it is unfolded, you will not get any signal, right? This is a monomer emission. It will get the signal, but it is in the form of a monomer emission. But when it form a complex with the potassium, result in the quadruplex formation. So potassium can form a complex in the form of what we have discussed here in the previous slide. So when this type of structure formation is there, potassium gets trapped and these two molecules come closer, and this would result in the excimer emission, and hence you can distinguish it. This is the one of the application. So for monomer emission, like we get 390 nanometer range in the absence of potassium, but when it binds to the potassium, the 480 nanometer band that has been observed. And the selectivity coefficient calculated for this particular sensor for potassium as well as sodium ion is much higher than the other sensors, because there are so many sensors which are being used for the determination of potassium and sodium ion in various samples. Then G quadruplexes as optical sensors for the proteins now. So you can see over here the lot of work has been done to modify the TBA, that is a thrombin binding aptamer sequence, to act as a protein sensor. So Hafiz et al. Were the first to report this by modifying the 5 prime end of go back the 5 prime end of this aptamer with a fluorescent compound, and labeling it with the aminosiloxane linker, and enabling all of these compound having a covalent attachment, or you can say the covalent attachment of whole of this oligonucleotide. So one is an end is a fluorescent compound, another end is the siloxane segment. So the covalent attachment of whole of this compound to the aminosiloxane linkage, which is mounted on the glass. So in order to sense the protein interaction by this probe, the fluorescent anisotropic technique has been used. So I will not go into that also. So this technique has also been discussed in the previous lecture. So this technique detect specific binding of thrombin in the solution to solution of the immobilized TBA ligand, as the protein DNA complex is much longer than the DNA probe itself. A significant change in the rotational diffusion ratio has been detected by the change in fluorescence and anisotropy. So this change is specific to both protein as well as for the thrombin binding aptamers. So no fluorescent anisotropy has been observed for the DNA sequence which do not form a quadruplex. So only when it is forming a quadruplex, you are getting the signal. When it is not forming the quadruplex, it will not show any kind of a signal. So these TBA sensors, they are very, very sensitive and rapid, and they could detect the protein, or they can detect the sample as small as from nanomolar to micromolar range within 10 minutes. So, very powerful sensor. Then there is another protein sensor. This has also been discussed, but I will just explain it. The Tan and his colleagues and some other groups use the aptamer back-on strategy to develop a real-time sensing thrombin. So again, the fluorescence resonance energy transfer as well as the excimer strategies has been used to study this kind of interaction. So how do these probe has been designed? So if you label this aptamer F at 5 prime end with the 6 FAM, and at the 3 prime end you attach is coumarin, uh, this one act as acceptor and this one act as a donor. This whole of this aptamer will prefer a random coil of conformation in a low salt concentration as well as or in the absence of thrombin. Right? I've discussed in the previous slides what is the function of thrombin. So, but equilibrium shifted when it senses the thrombin, and thrombin, what thrombin does is thrombin show the interaction with the G quadruplex formation. So these conformational changes will significantly enhance the fluorescent signal for 6 FAM, because upon folding, it is showing the interaction with the coumarin, that is acceptor donor kind of a interaction. So this assay or this kind of activity, they are highly sensitive to the detection limit of 100 nanomolar thrombin sample or molecule in a sample. Then G quadruplexes as a biosensor for the nucleic acid. So DNA can also be detected using the same thrombin binding aptamer, or you can say by interacting with this particular oligonucleotide. So group of scientists, they use the electrochemical version of molecular backend. Means there are two different approaches: the catalytic approach and molecular approach. So to detect the DNA hybridization by measuring the electrochemical signal that is being accompanied by a conformational change at the sensor level, right? This strategy is to attach the fluorescent tag to the thylated TBA sequence within a stem loop DNA structure, right? So in this case, in other approach, so I'll be discussing the catalytic approach. The thrombin protein was, this is the thrombin protein. This was covalently modified with the oligonucleotide containing TBA sequence. So this is thrombin protein. So we have an oligonucleotide sequence which is being attached to it. This is being modified. So in the absence of, this is the DNA strand. In the absence of the DNA strand, the TBA which is attached to this sequence will fold in the form of a G quadruplexes and blocks the enzyme activated site, so no further reaction will take place. Now, addition of complementary chain to this DNA fragment, which is being attached to this thrombin protein, enables, when the complementary strand forms a bond with this, or DNA strand bond with this, so this will not allow this quadruplex to form a complex, and it will further allow the or access to this thrombin active site, and will further proceed this reaction or allow the reaction to proceed, and we'll get a signal. So hydrolysis of this fluorophore labeled peptide, that would result into the detectable optical signal. So this is the way we confirm that which type of means this whole of this probe act as a biosensor for the nucleic acid. So, in my last slide, I will discuss what are nanomachines. Nanomachines, TBA sequence as a nanomachine. So, number of groups worldwide used a TBA sequence as a basis of a single molecular system to act as a nanomachine like this one, or a nanomotor. So you can see you have a G quadruplex. This is aptamer attached to this thrombin binding aptamer. This is being attached. So what is being done? The FRET analysis has been used to study the shrinking and expansion motion from aptamer to tetramer in a real time. So, conformational equilibrium between the DNA duplexes and quadruplexes define it as a nanomolecular machine. So, in this machine, as I told you, this is being fused with this TBA sequence was fused with another DNA sequence that can partially bind to the another sequence. So when you expose this to another sequence, the Q, it can partially show the duplex kind of an interaction, right, with this DNA at the second level. Upon addition of this DNA, right, so this QAPTB complex formation is there. So upon this complex formation, this quadruplex region that will unfold and it will release this thrombin binding protein, the protein which is bound to it. So addition of further, so this is being released, addition of another DNA sequence which is complementary to it allows it to refold again, right? And this machine, this nano machine represent or reported to be a new way to control the reversible binding of thrombin in a solution state. So with this, I finishes. Though I could not cover, there are a lot of interesting application because of the time and the scope. I could just confine myself to few important applications, specifically in the area of biosensor and cancer diagnostics. I think it might be helpful to you for your future endeavors in research related research areas. Thank you very much. And these are the references from where I have taken, and some of the references they are being are there at the as a footnotes on the slides also. So thank you very much. Over to Simran.
So, student have any questions? Any queries, questions?
I would like to say, if you don't have any query, it's okay because everything is online, or you can contact us, afterwards also. But all the lectures they are interlinked, so I would advise you just go through the previous lectures also, because terminologies, some of the terminologies they are common. For example, threat analysis has been discussed in detail, how this particular technique is being, used at an atomic level, at a molecular level. Then we have dimerization, excimer formation, Excimer emission, etc. So all these things has been discussed. So anyhow, if you have any questions, you can just write me and I will address that. Thank you very much.
Okay, thank you all the participants for listening the talks, and we'll take 15-minute breaks. Then we'll start the tutorials. Okay.
Yes, sir.